It's my greatest pleasure to introduce our second speaker, Chase Sweeter. Chase is a graduate student in Dr. Jay Shander's lab at University of Washington, where he develops technologies to study the regulation of protein degradation. As part of his graduate work, he created Comet, a high-throughput method for identifying A3 ubiquitin ligase substrates, which also incorporates a systematic evaluation of computational protein structure prediction for E3 substrate interactions. His research has been supported by NIH F31 award, and prior to his graduate school, he worked as a research tech in Dr. Jun Yang's lab at St. Jude and earned his undergraduate degree from University of Mississippi. And I guess he's gonna give a wonderful talk about Comet platform today. So Chase, now the floor is yours.
Okay, thank you. Yeah, I just want to say thank you to you guys for organizing this. So I'm in Jay Shinderay's lab as a graduate student, and this isn't a classical ubiquitin lab. So I found these seminars to be really insightful kind of as I've been a graduate student. But as the introduction kind of alluded to, I'm going to talk about this project that was recently published in Molecular Cell called Combinatorial Mapping of E3 ubiquitin ligases to their target substrates. And so this is just an outline of the talk. And I'll start off, even though I likely don't need it for this audience, with some background about ubiquitin-mediated protein degradation. So when I started graduate school, I kind of got interested in physiological protein degradation. And this starts with these E3 enzymes that conjugate this ubiquitin protein onto substrates. And these chains of ubiquitin are built up on the substrate proteins. And this can have two consequences. The protein can be rescued if the ubiquitin is cleaved off of the substrate by enzymes called deubiquitinases, or the protein can be targeted to the proteasome for degradation. And when I was starting graduate school, I thought that there were a lot of opportunities in ubiquitin biology, both from kind of just the basic biology of protein degradation, as well as therapeutic opportunities. So you could imagine stabilizing these interactions or kind of preventing them. And then also for variant contextualization. So for example, if you had a variant in an E3, if you didn't know what substrates it was degrading, you might not be able to kind of say anything informative about that variant. And then E3 substrate relationships are extremely complex. So there's about 600 E3s encoded by the human genome. And these are responsible for maintaining the protein levels of approximately 20,000 human proteins, not including isoforms. And when I was starting my graduate degree, it was very obvious to me that we kind of lacked methods capable of studying E3 substrate relationships at this kind of scale, right? So it's a very combinatorial problem. And so I kind of set out to make a dent in this issue, right? So I'll move on to talking about the method that I was able to develop. So this is called combinatorial mapping of E3 targets, or COMET. And this method relies on a paired E3 knockout and protein abundance reporter. So on a single plasmid, we clone a guide RNA that kind of using Cas9 goes and knocks out an E3 ubiquitin ligases. We have a DNA barcode, and then we have this GPS reporter, which was developed by the Elledge lab, where you fuse the potential substrate that you're interested in studying to GFP. And then there's this iris in-cherry bit that you can normalize the GFP signal to. So at the guide RNA position and the ORF position, we clone combinatorial libraries of both. So you kind of decide what E3s you want to knock out with the guides and what potential substrates you'd like to test. And then you clone all the pairwise combinations of them. So you end up with this barcoded combinatorial guide RNA ORF library. And the key thing here is that each plasmid, each individual molecule, carries a discrete genotype-phenotype pair. So the genotype comes from the guide RNA, and the phenotype comes from the ORF GFP fusion. And the workflow for this method is that you start with a pool of cells. In our case, we used K562 cells or HEC cells. You integrate them with the COMET library, and you get a pool of cells that have kind of a range of GFP intensities. Next, you use FACS to isolate them into 4 different bins based on the GFP intensity. And once you've isolated them using PACS, you can prepare sequencing libraries that kind of on the amplicon give you the guide RNA on one side and the barcode on the other side. And you do this for each of the 4 bins. You sequence this, and then you kind of look at the data to see if you can find any guide RNAs that are associated with shifting a substrate up a bin, right? So that would kind of indicate stabilization. And we also look at if we can find any knockouts that, for whatever reason, might destabilize a protein. So, we thought this was a cool idea, but we needed some proof of concept experiment. So we decided to focus on this really well-studied SCF complex. So this is a multi-subunit E3, and kind of the substrate specificity of this is determined by this green F-box protein in the upper left. And the Elledge lab back in 2008 identified 359 kind of likely SCF substrates. And they did this by using a dominant negative fragment against the Col1 scaffold. So, even though they were able to say that this substrate is related to the SCF, they weren't able to map what F-box protein was actually responsible for doing that degradation, right? So there's something like 70 F-box proteins in the human genome, and any of these substrates could have been degraded by any of these SCF F-boxes. So we thought this would be a good way to test our system. We cloned a combinatorial library targeting the F-box genes along with some core SCF components such as Col1 and Skip1. We used 30 SCF substrates from the 2008 Elledge paper, and then 62 non-SCF substrates as negative controls. And then for the guide RNA side, there were 73 genes that we targeted with 3 guide RNAs each. And we had 23 non-targeting control guide RNAs that we used to get kind of the baseline protein abundance. So we first looked at if we could measure baseline protein abundance. So this is, after doing the sorting and sequencing, kind of the read distribution for 4 different ORFs that we were like the least abundant in our assay. So these 4 ORFs, it's just showing kind of the read distribution across the 4 bins for each of them. And you can see that all of these have something like 80% of their reads falling in the first FACS bin, which is the least stable. And then at the other end, these are the 4 most stable ORFs from the screen. And so you can see each of these 4 ORFs have something like 80% of their reads falling in the 4th FACS bin. So these are the most stable, and it just kind of shows that we're kind of able to measure the baseline protein abundance, which indicates that we should be able to hopefully measure changes from the baseline. And so we calculate something called a PSI, which is a protein stability index. And this allows us to assign a quantitative value to each ORF paired with either NTC guides or targeting guides that knock out some E3, right? And so we compute this PSI, and then we clone some of these individually and measured the mean fluorescence intensity by flow cytometry. And what you can see here is that starting at the top of this plot, these go kind of low PSI to high PSI from the screen. And it also corresponds really well to the mean fluorescence intensity that we measure by flow cytometry. So the previous slide was looking at these ORFs paired only with NTC guides to see if we could get the baseline abundance. Now we wanted to look at these ORFs when they were paired with targeting guide RNAs. And interestingly, we were able to recover some known interactions. So this one is showing the PSI on the y-axis and the guide RNA target, either NTCs or when we knock out CCNF, which is an F-box protein, for this ORF SLBP. And this paper from 2016 showed that CCNF and SLBP are known to interact with each other, and specifically that CCNF degrades SLBP. This result was replicated over 4 independent transfection and sorting replicates. And importantly, this result kind of wasn't guide RNA specific. So this is showing all the guides that don't target anything, NTCs, versus all the guides that target CCNF, kind of faceted out. And so with all 3 of the independent guides, you see this shift in PSI. And then importantly, this validated kind of in a single experiment. So we just took cells, expressed SLBP, hit them with NTC guides or CCNF guides, and then you see that the SLBP GFP intensity shifts up quite substantially. We found this other example, which is TP53 being degraded by FBXW7, and the trends were very similar to the SLBP example. So we felt good that we were at least able to recover some known things, and then we wanted to go on and see what else was in the results that might not be so well characterized. And we also performed this screen in another cell line. So the previous results were from K562 cells, and then we set out to compare this to the same library screened in HEK cells. So this is an analysis that we borrow from GWAS, and it's called a QQ plot. And so you plot the expected p-values on the x-axis, and then the p-values that we observe from the screen on the y-axis. And I've colored the points by kind of what the guide RNA was targeting. So if it's an NTC guide, it's gray. If it's a targeting guide, but it didn't have a significant association, it's blue. And then if it's a targeting guide and it had a significant association, it's yellow. So if the assay is well calibrated, you would expect the NTC guides to track the diagonal dash line, which is what we see. And then we see an enrichment of kind of p-value signal in the targeting significant guides. And then we can plot this and look at the delta PSI. So what's the change in PSI when you knock out something, and compare that to the p-value. And then what I think is really interesting is comparing these results in 2 different cell lines. So here we're plotting the K562 delta PSI on the y-axis and the HEK293 delta PSI on the x-axis. And I would say that this is very directionally concordant. So if you're in the lower left quadrant in one cell line, you're likely in the lower left quadrant in the other cell line, and the same is true for the upper right quadrant. There are some, like in the lower right quadrant, that seem to be cell type specific. So there's a bunch of orange points in the lower right quadrant that are significant in HEK293 cells, but don't seem to have an effect in K562 cells. Right. So we felt good about this kind of small scale SCF focused experiment, and so we wanted to scale it up. So we decided to apply COMET to short-lived transcription factors. And this is paper Lee et al. From Molecular Cell in 2021, where they identified all human short-lived proteins. So we just took the intersection of the short-lived proteins from that paper and human transcription factors, and this gave us a COMET library where we had 108 short-lived transcription factors. And we want to see if we can identify the E3 that is degrading that, right? So instead of focusing on just F-box proteins, we shifted our focus to include all CRL E3s, as well as the APCC complex. So we integrate this library into cells and then do the sorting and sequencing just as we previously did. And we recovered this one really well-known example, PTTG1 being degraded by FCR1. FCR1 is also called securin. PTTG1 is also called securin. And then we found this interesting example where FCR1 is also degrading SOX9. And you can kind of group these by the substrate. So the plot on the left there is showing the substrate and how many things we linked it to. And then the plot on the right is showing the ligase and how many substrates it was linked to. Right, so overall, across these 3 screens, so there's the 2 FCF screens, one in K562 and one in HEK293 cells. And then the short-lived TF screen, we identified something like 103 E3 substrate pairs. And so this was quite a lot of pairs that we wanted to validate. And so we got to this point where we were wondering how to best validate these interactions. And I think the classical thing is to pick a handful of them and do experimental validation. But I think it can suffer from being biased, especially in terms of like intentional or unintentional cherry picking and reporting of results that work. Or another thing that we had thought about was using computational modeling as a scalable means for validation. So that's what I'll talk about now. So AlphaFold came out kind of early in my graduate school career. So there's this Nature paper from 2021, and then there's one for AlphaFold multimer that's, as far as I can tell, still on bioRxiv from 2022. And importantly, there have been demonstrations where AlphaFold multimer can predict E3 degron interactions. So there's these two papers from the Elledge lab that showed this. But kind of during the review process for this paper, we got a lot of pushback on how we were doing our AlphaFold predictions. And so we ended up kind of stepping back and saying, like, what is this missing and how could we be confident in the results? So what we set out to do was to do a systematic controlled evaluation of the application of AlphaFold multimer for predicting E3 protein interactions, as well as being as clear as possible for how we generated the predictions and how we report them. So this is how we did the prediction experiment. We had our 103 comet pairs. Those are the ones that we want to know if AlphaFold can predict the interaction. As a positive control set, we included 1,000 pairs from this bioplex data set, which is like a high throughput AP mass spec data set of proteins that should interact. And then we had two negative control sets where we predicted 1,000 comet negative pairs. So these were tested in comet, but not significant. And then 1,000 truly random pairs of proteins. So we run these through AlphaFold. We extract the prediction metrics, and we use this package called CollabFold Analysis from Harvard. And then we score the individual predictions and try to rank them. Right? And so this is what the results look like. If we start with our 103 comet-linked proteins, I'm showing here on the y-axis an AlphaFold metric called PAE, and then on the x-axis, this is another kind of metric for how confident AlphaFold is. And basically, if you're a good prediction, then you cluster in the upper right-hand part of this plot. When we look at the 1,000 bioplex predictions, there are definitely points that cluster up there, but which is encouraging because we think that this set should work. And in the screen negative set, you see that that cluster seems to drop out substantially, and most of the points are clustered in the lower left-hand corner of this plot. And this is true as well for the true random set. So we tried to combine the axes of these two metrics, so the PAE and the number of models per contact, into a single score. And then we just ranked the predictions and try to set a threshold for this score that best separates kind of the comet-linked predictions and the true random predictions. So on this plot, we draw this line, I think it was at maybe 0.45, and we keep all the predictions to the left of that. And when we do this and we look at what survives this threshold, it's kind of interesting. Like these comet-linked pairs seem to even outperform the bioplex data set, which I wasn't really expecting. And then both the comet-linked set and the bioplex set outperform the screen negative and the true random prediction classes. So I'll go over some of the interesting things that we found when we looked at some of the comet hits that seem to perform the best. So this first one that I talked about previously was PTTG1 being degraded by FCR1. So I'm modeling the E3 FCR1 here in blue. And when we looked at the AlphaFold prediction, there were two strong interaction points with PTTG1. And so I'm just showing those here. It's positions 1 through 15 and 58 through 67. And we did this really interesting computational experiment where we folded the substrate PTTG1 alone, and that's shown with this red curve. And then we folded the substrate PTTG1 with its E3 FCR1, which is the blue curve. And when you fold PTTG1 alone, the red curve is, the prediction metric is kind of middling throughout the whole prediction. But when you introduce the E3, you see these two sharp spikes right around these motifs that were shown to interact with FCR1. And this is just the ratio of these two metrics. And so we thought that these might be potential degrons, and it turns out that FCR1 is known to degrade PTTG1, and it's known to degrade PTTG1 using these two degron motifs at these positions. So we found that result to be very interesting and highly motivating. We then took this and validated it experimentally. So when we fused full-length PTTG1 to GFP at the N-terminus and either knocked out FCR1 or introduced NTC guides to the cells, we showed that kind of without FCR1, the protein is stabilized. We then took just the Degron regions. So I took from amino acid position number 1 to 84 and fused that to GFP. And we also see a significant stabilization. When we split the Degron in half, we lose that stabilization. So if I just take residues 1 through 42 of PTTG1, and fuse that to GFP, there's no stabilization. And also if I take 42 through 84, we lose the stabilization as well. And then we found this example of FBXO21 degrading APBB1IP. And this one was interesting because this was the only interaction that we found where it looked like the Degron was structural, like it had this helical structure to it. And there's a paper from the LH lab where they mapped Degrons that also found FBXO21 degrading a helical Degron, right? So kind of jives with what has been previously shown. And then experimentally, when we take full-length APBB1IP and fuse it to GFP and knock out FBXO21, we see stabilization. And if we take the Degron, stabilization seems to be even stronger. So that's kind of the results that I have. And I just want to talk briefly about how we might use these predictions to invert our current workflow. So what we did kind of in this manuscript is, we designed this library of guide RNAs and substrates that we want to test. We screen them, and then we use the computational prediction for validation, right? I think there's also a very realistic future where we lead with computation. So we make all of the predictions first, let it tell us what it thinks, is interacting, and then go and do some high throughput experiment to validate that. Right? So in this study, we predicted 3,103 structures, E3 protein interactions. This required about an hour per prediction, which is something like 157 GPU days. So what would a proteome-wide E3 prediction set look like? So if you had 600 E3s by 20,000 proteins, that's 12 million predictions. So if we just kind of said an hour per prediction, then we need on the order of 1,400 GPU years. So that's currently unlikely, but there are things that you could tweak to get towards this proposed future, right? So just to summarize the talk, Comet is an assay for testing possible E3 substrate relationships at scale. We computed these models of E3 protein interactions, and they seem to nominate known and putative Degron motifs. And then our systematic assessment of AlphaFold multimer points towards scalable in silico substrate discovery. And with that, I'll just kind of acknowledge my PI, Jay. So he's not an E3 person, but when I started graduate school, he gave me a lot of independence to try this project. And then along the way, I was mentored by Ning Zhang, who's also at the UW, as well as a postdoc in the lab, Diego Calderon, who recently started his own lab at UCSF. And with that, I'm happy to take any questions.